Okay, so now we're going to continue with our discussion of Chapter 8. And Section 3 moves into ATP, which is a very important molecule in living things, because you probably remember hearing it being called as the energy currency in living things. Indeed it is. You can see the type of molecule it is here. We have an adenine here, a nitrogen base, a ribose, which is a pentose sugar, 1, 2, 3, 4, 5 carbons, and some phosphate groups. And the conversion of ATP into ADP, breaking off one of those phosphates, releases some energy, and as we'll see here, this energy can be used to do work. So, for example, here we want to take glutamic acid, combine it with ammonia to create glutamine. So this is an endergonic reaction. It's a reaction that requires energy to carry out. Alright, we'll look down here. When we convert ATP into ADP, we release this much energy, 7.3 kilocalories. So we can couple this reaction so ATP can provide the energy to convert glutamic acid and ammonia into glutamine, because the breakdown of ATP into ATP provides enough energy to carry out the reaction. If this took 8 kilocalories or 9 kilocalories, a single ATP would not provide enough energy. Okay. ATP, again, is used to do lots of different things. Those transport proteins we talked about in the membrane chapter. Again, particularly to transport against the concentration gradient. It's going to take energy to do that. Those motor proteins that move organelle around along microtubules, it takes energy to do that. And so your living things are constantly breaking ATP down to release energy and then converting ADP back into ATP by taking in energy. And so essentially over here on this side we have respiration, cellular respiration typically, converting ADP into ATP, and then the ATP, the energy in those molecules is being used to do some kind of cellular work. Okay. Alright, the role that enzymes play here. We've talked about macromolecules and that proteins, one function they often have is in the form of enzymes. And again, they facilitate reactions. So here we have hydrolysis of sucrose. Now, that is, as we talked about, a spontaneous reaction, but it's a spontaneous reaction that happens very slowly. So sucrase here, notice that ASE ending, that gives you a clue. That's a tip-off. This is an enzyme. Facilitates this reaction. Now, I know this is something you probably talked about freshman year, but let's just cover it again, this activation energy. So again, we have this exergonic reaction. We start with a molecule that has quite a bit of energy, and we break it down into ones that have a little bit less energy, essentially making some of that energy available to do work. Alright, well, that spontaneous reaction has this sort of energy hill that it needs to get over for the reaction to go. Well, without the enzyme, that energy hill is significant such that it really doesn't happen. But the enzyme lowers this hill, this activation energy required to get the reaction going. You could probably think of it as even being somewhat lower. It just really allows that reaction to go by lowering the amount of energy that's needed to put it in, that's needed to get the reaction going. So enzymes essentially lower the activation energy. Alright, so enzymes, these proteins, they have a three-dimensional shape. They take substrates, act upon them to make products. And so what they have is in their three-dimensional shape, they have an active site, a particular spot on them where the substrate fits in there, what you would call the induced fit model. And then the enzyme, once you get it in there just right, is able to act upon it and carry out the reaction. Now, enzymes have sort of optimal conditions under which they work, and so that could be temperature or pH. And so here are two different enzymes. So the enzymes, or pretty much most enzymes in humans, are going to operate at your body temperature basically, which is a little less than 40 degrees Celsius. Whereas a bacterium that lives in a hot spring is going to have an optimal temperature for its enzymes. It's going to be significantly higher. And in regards to pH, again, most of the time your body fluids are around neutral or a little above that. Remember, your blood was, I think, 7.4. So most of your enzymes, including trypsin here, which is in your intestines, is going to operate at a fairly high pH, whereas pepsin, this one in your stomach that helps to break down polypeptides, operates at a really low pH, and so that's why your stomach pH is quite low, so that pepsin can do its job. Okay, enzymes, their function is sometimes inhibited. So you got the active site, the substrate would normally fit in there just so, and it would be acted upon. Well, sometimes you can have other molecules that get into the active site and essentially block it. These are known as the competitive inhibitors. Non-competitive inhibitors are compounds that attach somewhere else on the enzyme and change the shape of the active site such that the substrate no longer fits in there properly. It just occurred to me that this enzyme in this condition, it looks like it's given the hang 10 sign with someone's hand. I wonder if that was intentional. Okay, so that's enzymes. Now, section 5. The regulation of enzymes. So, of course, they can be regulated to a certain extent by the conditions they're in, whether or not they're going to do their job. But some other ways, you can have what you call allosteric enzymes. And allosteric enzymes are enzymes whose shape can change. They can go from active to inactive forms. And they're often found in these clusters. So you can see this cluster of protein subunits that are enzymes. Each has an active site and can carry out the reaction. And what can happen is these allosteric enzymes can be influenced by these things known as activators and inhibitors, such that the activator sort of locks in there and gets all those subunits ready to do their job. And the inhibitor, by simply locking into one place, can influence all of them. So, allosteric enzymes in these subunit clusters, all of them being either activated or inhibited. And here's an example where the substrate, which is being acted upon, actually sort of acts as its own activator, you might say, gets in there and makes all of those subunits active and ready to go. Okay, so another thing about enzymes and biochemical pathways. So here we have a biochemical pathway, and the substrate is this compound called threonine, and we're going to turn it into isoleucine. Okay. These are two different types of amino acids, so we're going to convert one to another. So you got a series of steps in this biochemical pathway, and an enzyme at each step facilitating the reaction. All right, so the goal is to make isoleucine. Okay, so let's say you have converted a lot of the threonine into isoleucine, so now you have lots of isoleucine present. Well, it would probably make sense that if you've got a lot of the product present, you don't need to make as much anymore. right? So what can happen is the product can serve as a feedback inhibitor of an enzyme, a protein, earlier in the process. So it will block the action of this enzyme, essentially slowing down or even stopping this pathway. Now, when later you're now short of isoleucine, there's not much of it around, this feedback inhibition decreases and the pathway is carried out. You should think of this not as an all or none process, but sort of a relative process, because you'd have a number of these enzyme molecules. And again, when there's lots of isoleucine, most of them would be blocked and not doing their job. When there's little isoleucine, most of them will be doing their job. So it's kind of a rate-dependent thing. It'll speed up or slow down based on the amount of isoleucine, the amount of feedback. And we refer to this kind of feedback in which you're slowing down the process as negative feedback. You are essentially negating the process, slowing it down. Living things operate often on negative feedback. For example, something like your body temperature, it starts to go up. You begin to sweat to cool yourself down. That's negative feedback. You're negating that trend of your body temperature rising. Okay, a couple questions, a couple things to ponder for class. Okay, so look at this. We've got a biochemical pathway that splits into two branches. Again, you would have an enzyme at each step facilitating the conversion of L to M and then M to O, etc. And so we've got several places here where we have negative feedback. So the question I have to you is, just by looking at this, does it make sense that Q would negatively inhibit the step between O and P, and that S would negatively inhibit the step between O and R, and that if Q were to go all the way back here and inhibit the step between L and M, that wouldn't make sense to you? Why does the scenario pointed here make more sense than if Q or S were to inhibit the step way back at the beginning? Okay, think about that. And here, so this diagram shows we've got time, the amount of product being made, and we've got a number of points on this graph. And so the question to you is, at what point in this reaction is the production of product occurring at the fastest rate? Okay. And on the other hand, at which points or point is production essentially slowed down or stopped? Okay, we'll talk about it in class.